We would like to welcome you to the Sanguine Speaker Series webinar, Precision Medicine for Genetic Disease, From Prevention to Treatment. Today's webinar is presented by Dr. Liao, the Laboratory Director and Assistant Professor at Columbia University Medical Center. And I will hand you over to Dr. Liao.
Thank you, John, for your kind introduction. So today, like you can see my title, I'm going to talk about the precision medicine for the genetic disease. I will cover from prevention to treatment. So yeah, this is outline on my today's talk. So first, I will try to answer question why we think is prevention medicine is also important for genetic disease. And then I will talk from diagnosis, prevention to treatment of the genetic disease. And last, I will use two very common genetic disorder. One is the cystic fibrosis, one is the spinal muscular atrophy, as an example to show how the prevention medicine can be practiced in prevention to the treatment of this kind of disease. So first, why genetic disease need a prevention medicine. So first, I want to make clear about the definition of a genetic disease, because in the broad term, probably every human disease have some genetic factor, right? So, however, the genetic disorder I discussed here, I more emphasize on the more narrow term, which is the monogenic, or someone called Mendelian disease. So this kind of disease, they normally have caused by the very rare, alleles, but their effect size is much larger. So normally it's caused very, dramatic effect to the, human health. So although individually, this disorder probably is rare, but collectively, actually, it's not that so rare. So for clinically recognized monogenic disease, actually, we can find in 0.4% of live births. But if you including all the congenital anomalies, it could be up to 8%. Also, over the entire lifetime, it's estimated that the prevalence of the monogenic disease is about 2%. So, so far, I think about 4,000 no human, gene can cause monogenic disorders. And traditionally, this kind of genetic disease gene was found by we call phenotype-driven strategy, which is normally have to classify the phenotype, and then the group the patient together as one kind of group of patient with a similar phenotype. And then we normally do the positional cloning. It's very time consuming and effort. We do the that's before the sequencing error, and then normally you have to use Sanger sequencing or other genotyping method to first narrow down the gene location by position cloning, and then do the Sanger sequencing to find out the mutation which caused the disease. But nowadays, start from I think 20 years ago, with the completion of the Human Genome Project and the development of the next gene sequencing technology, I think currently you can see the gene discovery is much more accelerated as shown in this curve. And also nowadays you can see by this blue bar here, majority of the new gene was discovered, to associate with human disease is found by the NGS technology. So this is kind of really change our strategy from the phenotype-driven to the genotype-driven, because we can just sequence the patient cohort and then find what possible novel gene could cause human disease. And that's very advantage for this strategy is some disease have a variable phenotype. So by the traditional method, you probably cannot classify them into the same category of the disease. But for the genotyping driven, we can easily find the similar mutations or the mutation in the similar genes. And also another advantage is a lot of this human disease caused by the de novo dominant variant, which is difficult for the traditional method to find the disease gene, but for the next-gene sequencing, we can just sequence the family tree, which including the proband, mother, and father, and we can easily detect this de novo dominant variants. So like when we talk about precision medicine, normally people probably first think of the maybe cancer, targeted cancer treatment, and maybe think about pharmacogenomics or some complex disorders. But actually, I will argue here, actually, genetics disease is really great area for us to practice the precision medicine. One reason is locus heterogeneity. So which means a lot of genetic disease is exactly the same disease could caused by the different genes. Like one of the example is hypertrophic cardiomyopathy actually is caused by more than 50 genes. And another disease, retinopathy, like I show here, it actually can cause around 20 different genes. And then another reason is also even maybe two individuals have the disease mutation in the same gene, but even within the same gene, it could have a lot of different mutations or variants could cause the disease, right? One example is CFTR, that's a disease gene to cause cystic fibrosis. We already found more than 2,000 disease-causing variant in this gene, and a lot of this variant actually is very rare or it's private, so it's probably just one individual carry this mutation in the whole world. So that's obviously need individualized treatment. So the next, because also because the genetics or environmental modifier, so even the different individual could have a different different severity of the phenotype. So we normally in genetics we call the penetrance and the expressivity. So it could have a variable expressivity or incomplete penetrance. So that's also you need to individualize the treatment for different phenotype. And last, I will say I will argue that the basic research in the genetic disorder, because normally the genetic impact is very strong in this genetic disorder, so it's very easy to find out some very significant biological pathway, which important for even for the common disease. One of the good example is familial hypercholesterolemia. So this is a genetic disorder caused by a gene called LDL receptor gene, and normally if you lost a function of this gene could cause this disease. And later, I think people found actually a reagent called statine actually can increase the expression of this LDL receptor. That's why they can decrease the cholesterol level in the human blood. So that's this pathway found by this genetic disorder actually can help us to discover this new treatment drug. The statin currently is very common drug to use for treat the patients with high cholesterol. So next, I will talk about the genetic disease diagnosis. As you can see, the first, before we can go ahead for treatment or prevention, we first need to make a diagnosis for genetic disease. Historically, I think the very basic, the first whole genome test in the history, I think is this chromosome karyotype, right? So basically, if you look at this, if you look at this picture, you are seeing the whole human genome, right? But it's just like the resolution is so low. So probably the only thing we can say is that we can count the chromosome number, right? So we can tell the genetic disease caused by the gain or lost chromosome. For example, trisomy 21, we know it's caused Down syndrome, or monosomy X caused the Turner syndrome, right? So the later with the development of new technology, we have this tool called microarray, right? So you probably know the microarray will detect the large deletion or duplications in the genome, normally from several hundred KB to several MB, right? So this one actually will have a better resolution, but still it's only a small portion of genetic disorder can be detected by microarray, some microdeletion like duplication syndrome. You probably heard of, for example, the DiGeorge syndrome or Prader-Willi syndrome, Angelman syndrome, right? That's all caused by the deletions. And the last, I think, with the development of next-gene sequencing technology, eventually we can have the best resolution to look at the human genome, which getting to the single base pair level, right? So, however, the challenge for this, I will talk about in detail later, is although they can dramatically increase our detection rate for the genetic disorder, but at the same time also they give us many variants we don't know how to do with it, you know. So that's also a challenge for that. But still, with the application of the NGS technology in the diagnosis field, you can say right now we can get relatively good diagnosis rate for genetic diseases. Like this show, like in general, the diagnosis rate is from 30%, for example, for developmental delay, to the highest category is retinopathy. You can get 70% of the diagnosis rate. So the question is, again, why we still cannot get near 100% diagnosis rate? Why is still a lot of patients, they cannot get answer for what kind of genetic disease they got, right? So they have several, I call challenge or the reason for that. So first is technical limitations. Currently, you know, the short reads-based next-gene sequencing still kind of difficult to find variants in, for example, non-coding regions or some structural variant like a balanced translocation, and also some disease caused by the repeat expansion, or some gene have a pseudo gene. It's all kind of challenge for current NGS technology. And the second, you know, like I said, I mentioned earlier, only like around 4,000 gene we know it's cause disease, right? But in the whole human genome, we have over 20,000, right? So about 80% of gene, which we still don't know what's their function. So it's still not a lot of new gene. It's need to be discovered. And the last, I think, like I mentioned, this next-gene sequencing will give us a large amount of new variants, right? So a lot of these new variants, we don't know what's their clinical impact. So normally we call this variant of uncertain significance. And so that's like we don't know how to do with it. We don't know what's their functional impact to the disease. And also like sometimes if you run the genetic test in different labs, and they may report, even for the same variant, you could report as a different category. One lab could call it a variant uncertain significance, but other lab could call it likely benign or benign, right? So they still have a lack of the consistency in the diagnosis field. So there's some potential resolution in the horizon. And I think one is for technical part, besides the short reads, whole exon sequencing. Not a lot of labs right now, they start to offer clinical like whole genome sequencing. So for the whole genome sequencing, they can cover these non-coding regions. And also some new technology like, for example, this long-read sequencing like a PacBio or Nanopore, they can sequence at very long length. So that can also detect the structural variations and also some repeat regions. For the the novel disease gene discover, I think one of the effort in the field is push the data sharing. So it's a lot of website or database is built for that purpose, like a matchmaker exchange, ClinGene, ClinVar, Nomad. It's all very useful for the gene discovery and variant interpretations. And last, I think is some research which try to do some high throughput function study to help us to interpret the functional impact of the variant we found by NGS, right? So one of the good example is one of the recent paper published in 2018. This paper actually they use gene editing method to basically make a mutation for every possible nucleotide in the functional important domain in the BRCA1 gene, right? This gene, it's caused familial breast cancer, ovarian cancers. And then basically they try every possible mutations could possibly happen in the human. And then they use some high throughput functional in vitro assay to see how this mutation affected the functions, right? So this kind of study is really, really helpful for the clinical interpretation for this for the variant we can find in the human. So another thing about the variant interpretation is this the guideline was published several years ago by the American College of Medical Genetics and Genomics and also Association for Molecular Pathology. So they try to standardize the variant classification. So they propose that based on the evidence strength, they classify the variant into 5 categories: pathogenic, likely pathogenic, unknown, likely benign or benign, right? So most labs currently, I think, follow this guideline. So currently, we actually see more and more consistency between the report from different laboratories. So next, I will move from the diagnosis to prevention, right? So like one of the main point for prevention, precision medicine is we should shift our focus from a reaction to the disease to prevention of the disease, right? So because the best strategy to treat the disease actually is not the treat the genetic disorder after it happened, right? It should be prevent it from happening, right? So that's why I think the genetic disease prevention is really important, for the precision medicine. So actually, genetic disease prevention can happen in multiple stages across lifespan, and at the earliest stage, like for example, in the conception stage, I think we can run the carrier screen, which find out the potential carrier couple, right? And then before or after pregnancy, we have some other methods such as Ph.D, prenatal testing or NIPT can find the fetus or embryo which have carry the genetic disorders. And then after baby was born, like we can have a newborn screen, and we can prevent some genetic disease, which very easy treatable from some supplement or from like a dietary restriction. So that can prevent the onset of the certain genetic disorders. And eventually, some genetic disorder is adult onset, right? So, for example, we have some we call the predictive testing, like for example, the BRCA1, BRCA2 gene test for the woman with family history of early onset breast cancer or ovarian cancer, right? So that's all like a predictive testing. And also when we run the diagnosis test for the family, we also report, we call the secondary finding, which means some mutation which not the reason for the original indication or diagnosis, but it irrelevant to original patient symptoms, but it could have some later onset disorder, like for example the increased cancer risk or cardiovascular cardiomyopathy disorders. So for that one, we also report to the families. So this is also can prevent it from happening if we have some proper medical intervention. So here, the rest of the talk for this section, I will most focus on the carrier screen, because you can see one reason is this is a first step of the prevention, right? So obviously, it have the best impact to the prevention of the genetic disorders. And the second, actually the genetic carrier screen is become very, very popular recently, and it's probably one of the most common order test, for genetic lab. So that's why I think we should look at a little bit more on this carrier screen. So for traditionally, carrier screen is only offered to a certain ethnic group or race which have a high incidence of certain genetic disorder because of the founder effect or the carrier advantage or some consanguinity happen in certain culture. And for that one, normally, like I mentioned, because the limitation of the screen method, normally we cannot even sequence the whole gene, right? So normally it's only limited to a certain number of founder mutations and in a single gene, right? So that's a kind of a limitation for this traditional carrier screen. However, regardless of its limitation, it still have a huge impact in the public health. This is a good example for the traditional carrier screen. And one is the beta thalassemia in the Sardinian, and another is a Tay-Sachs disease carrier screen in the North American. So you can see the both carrier screen start around like same time around 1970s. And then you can see the incidence of these two diseases in these two populations is very high. But with the carrier screen was offered in the whole population, you can see they have a dramatically decrease of the disease incidence, right? So actually, one of the good control is this red curve here. This is show the incidence of Tay-Sachs disease in the non-Jewish population. And at that time, they are not being offered of the Tay-Sachs disease carrier screen. So you can see their incidence is not changed. And actually nowadays, it didn't show the current data, but nowadays, actually majority of the Tay-Sachs patient is actually non-Jewish, although this Tay-Sachs disease is originally considered as a Jewish-specific genetic disease. So the medical society, they have some common suggestion for some very common genetic disorders should be offered for the carrier screen. And you can see from this table that including cystic fibrosis, spinal muscular atrophy, Fragile X disease, alpha thalassemia, beta thalassemia, sickle cell, and then they also offer the Tay-Sachs, some other disease in the Ashkenazi Jewish population or the French Canadian populations. Recent year, actually, they have another test offered more and more common in the carrier screen field, which is called expanded carrier screen. So compared with a traditional carrier screen, the expanded carrier screen is they can simultaneously screen a large number of conditions at the same time. And also normally they don't pay attention to what racial or ethnic background, so they can screen any general populations. And the reason why this new test was emerged for the two reasons. One is the advance of the molecular technology, like majorly is next-gen sequencing, right? So basically we can sequence the large amount of the genes with the same cost, even lower cost compared with the traditional carrier screen. And the second, more and more individuals nowadays actually born in the family with multiple ethnic background, right? So it's more difficult for nowadays people define themselves as a single ethnicity, right? So that's another reason it's become more popular, this pan-ethnic expanded carrier screen. And one of the advantage obviously is can increase the detection rate, right? So you can see here, if we use the disease incidence as 1 or 100 as cutoff, we need the test 18 conditions at the same time, and we can detect 61% of carrier and 84% of at-risk couple. However, if we increase the, or actually decrease the incidence cutoff to 1 in 500, then we need a test 91 condition at the same time. However, that's going to be increase our detection rate to the 80.5% for the carrier and 95% for at-risk couple, right? So obviously more disease, more gene you tested, and the better detection rate will be. However, one of the caveat you can see is with the more gene added into this expanded carrier screen, eventually this curve will reach to a plateau, right? So which means, if you add more and more like a rare, the genetic disorder have very, very rare, right? We are not going to increase the detection rate any further, like, at least not dramatically, right? So that's why, these have some potential issue for current expanded carrier screen. One is, because we're doing the sequence, next gene sequencing for a lot of genes, right? So like I mentioned earlier, we'll get a lot of variant. We don't know how to do with it. This variant on certain clinical significance, we don't know how to interpret it, right? And the second, also because more disease gene we tested, we eventually like more than half of the individual will become a carrier for certain disease, right? No one is perfect. We always, have some genetic the defects in our genome, right? So they will increase the genetic counseling, effort burden to the limited number of the genetic counselor workforce. And at last, as I mentioned, because currently it's market driven, right? So it's a lot of companies, they just try to add more and more genes and conditions into their carrier screen panel, but eventually they will reach to the plateau. So currently, I think it's need some regulation in which gene should be put into this carrier screen. And so next, I will move from the prevention to the treatment, right? So eventually, if we have the patient with a genetic disorder, then what we should, what's the best treatment strategy? Traditionally, you can say we can treat this genetic disease with at different level. They have a different strategies. And the traditional treatment normally is at the lowest level, like most downstream level, which in the phenotype level, right? So they basically just treat the phenotype of the patient. One of the example is, for example, if a patient have a thalassemia, then they will give the transfusion. And then, if a patient have done some congenital heart defect, they will have the surgery to fix the heart, right? So this is just in the phenotype level. I won't go to the detail, today. That's not the focus of today's. And however, once we move to the more upstream, then which we need to know better about the disease mechanism and what gene and what mutation to cause the disease. And then we really can treat them individualized, right, treatment, and that go more into the precision treatment area. So, so that's why the next level, we can treat the patient in the pathway level or protein level. So this is several good examples. So for the pathway level, some of the genetic disorder affect the pathway, right? So because the genetic defect, this pathway cannot go through, then they have accumulation of the substrate, right? So one of the pathway treatment, at the pathway is a substrate reduction, right? So then we can get rid of this substrate. Or we can, maybe we call pathway inhibitor, like in number 2 here, we can inhibit these pathways to cause less severe disease phenotype. And this is all in the pathway level. And however, in the protein level, we have also different strategy, but this required, we know what kind of mutation, what's the effect of the mutation to cause the protein. For example, in number 3 here, so this mutant protein, actually, they cannot transport to the, for example, the cell membrane where they can have a proper function, right? So then if you have some small molecular drug which can bind to this mutant protein, and they can help the transportation and then maybe you can restore the function of the protein, right? So this kind of a small molecule drug called the corrector, right? So the number 4 is another for another mutation, which, although this mutant protein can transport to the cell membrane, but they are not have enough function, right? So they have another type of a drug called potentiator. So this one can bind the protein and increase their function, So and last, some protein maybe they have a stable stability issue. So maybe they get degraded too early. So some small molecule we call like stabilizer, which can bind to this protein and make it more stable, right? So all these different treatment at the protein level, we need to know what kind of mutation it is, and it's more individualized treatment and more into the precision medicine area. And eventually, like if we know better about this, we also can treat disease in the RNA level, right? We can change the RNA expression, and we can change the RNA splicing. One of the very important tool for the RNA level treatment we called antisense oligonucleotide. So this antisense oligonucleotide normally is about 15 to 30 base pair, and they target to the certain sequence of the gene, right? And normally the backbone of this oligonucleotide is modified, so it's more stable in vivo, right? So they can depend on your design and where they harbor to the mRNA, they have a different function. For example, if you can just bind to some important exon, right, then you can introduce RNA's cartilage. Then you can degrade this mRNA, right? So that's a pretty basic function. And sometimes you can design this antisense oligonucleotide to the translation start, AUG side, right? So when they bind to this side, they can, inhibit the binding of the translation machinery into the starting code, and so they can cause translation arrest. And also, some mRNA binding protein can bind to some certain non-coding area, and they cause some cellular toxic, effect. And if you use this antisense oligonucleotide to bind to this RNA domain, and so they can inhibit the RNA binding protein to bind to this RNA and can reduce the toxicity of this binding protein. And also, if you design the antisense oligonucleotide into the intron exon boundary, they can also change the splicing of the mRNA, so you can cause exon skipping, like for example here. And also, if you design this antisense oligonucleotide into the upstream of the open reading frame, actually you can increase the gene expression for the downstream open reading frame. So you can increase the translation activities. So it's a lot of usage for this antisense nucleotide. And this is actually two good examples, which actually it's I think both are FDA approved drug, which is use the antisense new oligonucleotide to treat the genetic disorder. One example is they call eteplirsen for the Duchenne muscular dystrophy. So this Duchenne muscular dystrophy is caused by lots of function mutation in this gene called DMD, right? So you can see they have a lot of frame shift, a lot of function mutation, for example, frame shift here in these genes, right? So this antisense mRNA, they can actually bind to the intron exon boundary of this exon 51, right? And then they can make this RNA splicing actually skip this exon 51, right? So once they skip this exon, like sometimes they can reinstall the open reading frame, because a lot of this other mutation is caused the frame shift, right? Then they cause the non-functional protein, which is eventually degraded. However, this exon skipping of the 51 for about 13% of patients, in 13% of the patients, they can restore the open reading frame, right? So the final result is you're gonna get the shorter protein, but it's in frame, right? So they still have a partial function. So they still can, kind of function, restore some function of the compared with the loss of function mutation in here, right? So that's one of the treatment for the Duchenne muscular dystrophy. Another example to use this antisense oligonucleotide is this drug called minomycin. This is for treat the hypoglycemia, hypoglycemia. And this one is caused by the gene called the ApoB, right? So this ApoB, I think they actually is an important component for the LDL in the bloodstream. So this antisense oligonucleotide can bind to the pre-RNA of the ApoB gene, and then they can, introduce the RNase-H, and then they are going to have a cleavage on this mRNA, and then which cause the decrease the expression and the ApoB gene, and eventually decrease LDL level in the bloodstream. So finally, I think we can move to the highest level in the gene level, right? So eventually, this is the final goal of the genetic disease treatment, because potentially they can treat a genetic disorder in the highest level and have a potential to cure the genetic disease, right? So this one, for certain disease, we can do this by the transplantation, right? Organ transplantation, for example, the bone marrow transplantation, or some, currently use stem cell for transplantations. However, I think more interesting development in this field is gene therapy and gene editing, right? So for gene therapy, I think the basic concept is very straightforward. It's just because a lot of this genetic disorder caused by loss of function, in the endogenous gene, right? So if you introduce a transgene into the human body, and then they should restore the function, right? So, however, I think the challenge is how to deliver this gene into the human body. And you can see they have several different methods. I think the most common used method is this vector, transfer vector. The original come from virus. One of the most popular one is this lentivirus. So this lentivirus vector, I think advantage for this is relatively large. So you can put like up to 8 kb DNA fragment into, you can pack it into this virus vector. And however, the disadvantage for this vector is actually this vector could integrate into the chromosome, right? So potentially they could have some safety issue, cause a mutagenesis. And another type of common use is called adeno-associated virus. So this virus, I think they are relatively safe because they have a low frequency to integrate into the chromosome. And so their safety is good, but the problem is they can only impact like about the 1.5 kb maximum of the DNA fragment into the vector. And also for delivery strategy, also have two kind of delivery strategy for gene therapy. One is in vivo gene therapy, so which is by just directly inject this the virus vector impacted these genes, and then inject into the human body. And for this strategy, I think normally we use the adeno-associated virus. And the major risk for this strategy is immune response. Another strategy we call the extra vivo gene therapy. So this one, first we extract the cell, from the patient or from healthy individual, and then we culture them in the petri dish, and then also inject, put into the virus vector, impact with transgene into this cultured cell, right? and then then we deliver this genetically modified cell back into the human body, right? So normally the lantinovirus used for this strategy more often, and obviously one of the risks is you could have a mutagenesis, for this strategy, and also you need to have some cell, the cell type you are used have to be easy to take out from human body and put back in. So at last, I think the final is like a gene editing strategy, right? So this is a very popular, nowadays, a lot of, I'm sure everyone heard about this. It's a lot of publication about this gene editing, right? So the basic concept, I won't go to the details because, it's very popular topic here. But basically idea is we use this nucleotides called Cas9, and they can, with the help of this guided RNA, and they can recognize specific target sequence, and this Cas9 nucleus can cut the DNA strand, create a double strand break, and then in the downstream, either this break can use the non-homologous end join to join together again. But in this process, you will create insertion or deletions. So basically, you're gonna knock out this gene. Or if you provide some the single strand DNA donor template, and then they can, based on this template, and to fix the repair, they call the homologous directed repair. So they can repair this double strand break, and during this process, they will introduce a new DNA or new variant into this location. So this is normally could cause a locking. So basically for this strategy, they could also have an actual vivo or in vivo strategy. And currently they have several clinical trial is still ongoing. So it's still, hopefully in the near future, we can hear more progress on this. But I think that just early this year, they have already one paper published from, I think, one of these the first clinical trials. And this is like a paper published in New England Journal of Medicine. And actually in this paper, they only report two patients, actually. One patient have a sickle cell disease, another patient have a beta thalassemia. So basically they use this CRISPR-Cas9 gene editing strategy. They make this protein because for both of these patients, because the defects of the hemoglobin protein beta, right? So this hemoglobin beta is most expressed in the adult. However, in the fetal stage, actually human have another the beta, it's kind of like can replace the beta protein called the gamma protein, right? But this protein normally is not expressed in the adult. So they use the gene editing strategy and make this gamma protein expressed again, right? So then they found actually more than a year later, they found actually a high level of this the gamma protein in the patient, and also they found this patient have a transfusion independency, and also elimination of the episodes for the sickle cell patient. So it looks like the preliminary data is very promising. So even only in 2 patients, they can publish this paper in New England Journal of Medicine. So I believe like eventually we're gonna hear more about these clinical trials. So last, I think I will go to some, I will go to two specific genetic disorder, which I think is used as an example for the precision medicine. One is cystic fibrosis. So for cystic fibrosis, I think it's the most common autosomal recessive condition in US, especially in the Caucasian population. The carrier frequency is the very highest, 1 in 25. And obviously it's a complex multi-system disease, and most affect the pulmonary disease, but also they have some other organ get affected. And so the gene responsible for this disease called the CFTR. So they encoding CAMP regulated coronary channel protein, which is located in the apical membrane of the epithelial cells. And currently, more than 2,000 mutations have been identified this gene. And based on their functional impact, they have 6 different mutation classes. And one of the most important mutation is this deletion of the phenylalanine in position 508. Normally, we call it the delta 508. So it found in 30 to 80% of the patients, and this belong to the class 2. So they cause the major blocks in the protein maturation. So like based on which mutations you got, right? So you could have the phenotype, very classic phenotype, or you can have a milder phenotype. And to prevent this cystic fibrosis, even start at 1997, NIH already proposed to offer the cystic fibrosis carrier screen for certain populations. And in 2001, ACMG and ACOG, they have the guideline recommend this cystic fibrosis carrier screen offered to the non-Jewish Caucasian or African Jewish populations. And in 2004, ACMG recommend these 23 most common mutations they think should be tested in the cystic fibrosis. And nowadays, I think, with expanded carrier screen, almost every pregnant woman or the woman who consider pregnancy was offered for the carrier screen for cystic fibrosis. And this is the 23 most common mutations recommended by the ACMG. So all these mutations have an allele frequency of more than 0.1% in general US population. And however, like you can see, one of the issues for the carrier screen of cystic fibrosis is they have a different detection rate in the different ethnic group, right? So in the HA population or non-Hispanic white, the detection rate is pretty good. However, the Asian population is less than 50% of the detection rate. And the reason is you can see, in the East Asian population, you can see the top 3, candidate, most common mutation found in East Asian is not even in this 23, mutation list recommended by the ACMG, right? So, for the Southeast Asian, also like top 2, it's not that. So that's why I think here, I think the NGS technology compared with the genotyping-based technology have their advantage. One of the study published in 2020, they suggest that compared with NGS technology, the genotyping technology tests only 23 most common mutations, fail to detect probably 30% of the S3 at risk couple for cystic fibrosis. And you can see the NGS technology can detect most of the mutations in the East Asian or in the Hispanic populations. And so for the treatment, I think one of the very important treatment is this small molecular treatment, depend on the class of the mutations. And like I mentioned, for delta 508, the corrector plus potentiator is very useful to treat the patient carry these mutations. And this is 2 papers published in 2017, I think, and they combine these 3 drugs together. 2 of these 3 drugs are the corrector, and the last one is a potentiator. And they found in combine these 3 drugs, you really can have a very good treatment outcome for this any patient with at least carry a single allele for the delta 508. So this is the FDA recommend treatment. You can say the last one is this 3 combination of these 3 drugs for the patient with at least one delta 508 allele. This is really going to be helpful for the CF patient because you can see more than 80% of patients could carry these delta 508 alleles. Yeah, I think the last maybe 2 minutes, I will just briefly mention this spinal muscular atrophy. So this one, I think, is also second most common autosomal recessive disease, but it's a leading genetic cause of infant death. And they have 2 genes, SM1 and SM2, right? SM1 is a major gene contribute, to this disorder, then they have a 100% functional protein. But SM2, because of one of the mutation in exon 7, so it's only 15% of the transcription can have a function protein. 80% of the protein, they don't have the exon 7, and then they don't have a function. Yeah, so actually they have a different types of the SMA, and although SM2 don't have the major function, but the copy number of SM2 can modify the disease phenotype. You can say if you have a more SM2 copies, actually you can see some a milder phenotype in the patient. And so for the carrier screen, I think one of the issue for SMA carrier screen is this called the silent carrier. So the silent carrier, they have a 2 SMA gene in the same allele, right? So when we look at the copy number of SM1, we saw they have a normal as a normal individual, but actually they have another allele is missing a copy number of SM1. So this is called the silent allele. That's caused we cannot detect all the carriers in the SMA genes. And for the therapeutic, I think it's 2 very promising. One is again use this nonsense. So oligonucleotide, antisense oligonucleotide, they can target the exon 7 of the SM2, right? So normally the SM2 exon 7 cannot be spliced, but this they can modify this and change the splicing that make SM2 produce the normal function of the protein. So that's one good strategy. Another is gene therapy. So they deliver this SM1 gene into the patient, and then they can also have improved outcome for the patients. So I think that's it. So I won't go to the summary because of time, but I hope today's talk I can convince you that genetic disorder is really an important field to practice precision medicine. And I believe in the future with better understanding of genetic disorder and more new method for the precision medicine, we're gonna heard more new therapy or new prevention method for the genetic disorders. Thank you. So I think I can take questions here. So one of the questions, should we to apply the personalized medicine approach for more common form of the disease, right? What's required scientifically? Yeah, so that's, I think, is a good question, because normally more common form of the disease is much more complicated, because there's a lot of factors to contribute to it, right? So I think we need to understand better about the genetic mechanism, or maybe not only genetic, even come from the environment, right? So that's why I think, currently, one of the, in the diagnosis field, one of the very popular tests, currently called the polygenic score, so this is basically measured on different polymorphism from the different gene in the different pathway to try to calculate the risk for the disease, right? So I think that's just the first step. I think eventually with a better understanding, hopefully we can apply more personalized medicine to this common form of disease. So another thing, question is, have patients expressed concern with privacy of their genomic data? Yes, I think that's definitely a good concern. Currently, when we enroll a patient for their studies or for even the genetic test, right? So, however, I think normally we will provide a very very detailed, informed, consent, because we're gonna basically have the option for the patient whether they want to their DNA, not only DNA but the genomic information to be stored, or whether they want to contribute to the research. If they not agree to store their genomic data, we will, it's just within the limitation of the regulation requirement. After that, we won't store their data. And also all our like a database, everything is like a HIPAA, follow the HIPAA rules. So it's very data security is top our concern. Please stress another one, privacy protection for subject giving DNA sample, right? So that's a kind of same question as the previous one. Like I said, it's different level, right? Even in the DNA level, right? Normally we're gonna throw the DNA without their, if they are not agreed, we keep their DNA. We will, normally destroy the DNA after, I believe, the 60 days, after the test was finished. And for their genomic data, I already mentioned. What's some challenge, within the applied personalized medicine approach? Any question on the back to practice and implementation? That's, I think, also, very good question, because it's exactly mentioned, one of the challenge, I think, that because, all this, especially the treatment, I think is very, very currently all the treatments very, very high cost, right? So because it's individualized, right? So it's really normally it cannot practice in the large scale. I think that's the most challenging one. And I feel to address this, I'm not sure what's the best solution for that, but I think one of them probably, like I mentioned earlier, maybe we should switch from the reaction to the prevention, right? So I still feel the prevention is a better, it's better than the treatment when the disease already happened. In the prevention, you can have a much, lower cost effect, compared with the treatment. Are there sequences achieved, such as future genetic discovery can be applied? I'm not sure what this means. And when will precision medicine come to non-oncology application, right? So, yeah, I think, that's exactly the point of my talk today, right? So I feel it's, still it's getting kind of ignored, this precision medicine in the oncology, like all the today's talk is the non-oncology. I think the oncology is still the, I think the, more hot topic, for the precision medicine. But I hope, with more and more, people realize, this, in the non-oncology, genetic field, also with development of more new tests, hopefully, we're gonna draw more attention to this field. So I think that's probably the best, conclusion for today's talk. I think we already at the time. So thank you for everyone.
Thank you, Dr. Liao, for an excellent presentation. And thank you for joining Sanguine for our S3 webinar on precision medicine for genetic disease from prevention to treatment. For our list of upcoming webinars or to request patient samples, visit sanguinebio.com. Thank you again and enjoy the rest of your day.